Right. So hi, everyone. It's very exciting to be here. So my talk, I'm from the psychology department, so I'm a neuroscientist. So my research is more like, well, it's in the psychology. And my talk today is like how Garcia is helping me with my research, basically. I don't have a background in computer science. So it's very exciting to be here sharing what I do and how I got help from Garcia. So my talk is accelerating behavioral neuroscience with supercomputing and deep learning. So a little bit about me. I'm currently a postdoctoral of research in the Department of Psychology. My interest, and I just noticed it cut a little bit there, but that's okay, is in neuroplasticity, learning and memory processes, molecular neurobiology, and computational neuroscience. So in part of our research, we do a lot of prenatal alcohol exposure, substances exposure, pharmacological drug testing, with the purpose of understanding how those things affect memory and the learning processes. So when I was a brand new graduate student, this was in the fall of 2019, I had some projects that I was going to work on for my dissertation. And then there was a project in the lab that would be more like a side project. I was the only new graduate student there. So my advisor asked me if I would be interested in doing that. So I was like, okay, why not? I was a brand new student. I was eager to learn whatever was available. So he explained me the task, and it's the novel object recognition task. So very briefly, because I don't know if any of you has like a background in psychology or anything that involves in my talk. So basically, it's one of the most basic memory tasks that we do. We put the animal in a box with two objects that are the same. They will explore, get used to those objects, and then we wait 2 hours or 24 hours, depending on the protocol. And then we put the animal back there with one of the objects that they saw before and a new one. So basically, if the animal spends more time exploring the new object, it means they recognize that something was already familiar and there's something new there to be explored. So I do mice and rat research. They're very curious, and so if they're a normal animal, they will explore what's new. So because we do like drug tests and things that will affect memory, if they have any kind of memory impairment, they might not be able to recognize that there's something new in the environment, or they might treat the old object as a new one. So sounds very simple, like put two objects, put the animal there, record it, and then do a discrimination index, which is basically like the time that the animal spends exploring the novel object minus the familiar object divided by the total time. So when I heard that, I was like, well, that's pretty simple. It's something that, I didn't find anything challenging. The problem came when I asked, how? How do you time it? What do you do? Because, this is me talking with my advisor, and I was like, how people time it, the amount of time the animal spend exploring the object. And basically the answer I got was the least satisfying answer ever with a stopwatch. So basically they said, it was like, okay, people will record the animals, sit in front of a computer, play the video with a stopwatch, and they're tracking one object, watch the video again, with stopwatch, track the other object, keep track of time, and then that's it. Then I read the grant. We had 11 experimental groups, 20 animals per group, 4 videos per animal. That's 880 videos. The training, which is like when the animals are getting familiarized with the pair of objects, can take anywhere from 3 to 10 minutes, because it depends on how the animal is moving. The second one is 5 minutes. So I'm there as a brand new graduate student doing the math in my head, and I was like, assuming that I could do both objects at the same time and perfectly, that would take me at least 75 hours of scoring. And because of fatigue, reaction time variability, and human error, we need to have at least 2 scorers, and they need to be blind to conditions. So it would be like 2 people scoring 880 videos. So I wanted more. And I was like, well, the task seems pretty simple. The analysis of it is not interesting at all, even though you can say it's the most basic thing, sit with a stopwatch. And I was like, cannot do that. I'm not doing it. So I started talking with some researchers from other universities to see what they were doing, what's available, if there's something that was not manual. And most of the answers I got was, we always did it this way. And I was like, okay, we need to change that somehow. So there are some softwares that can do that, but they are too expensive. And plus, when you buy a software from a company who will design the software for that task, they normally design that for that one specific measure. If you want to do other measures, you need to buy other softwares and maybe, so you have the constraints that you don't have when you're using like an open source, something that you can program and manipulate. So around that time, this was end of 2019, a friend of mine introduced me to this paper. And again, it's important to remember that I don't have a background in computer science. So deep lab cut, markerless pose estimation of user-defined body parts with deep learning. I read the paper. All these words were there and more. And I saw all of that and was like, I have no idea what these things mean, but this is exactly what I want to do. So that sounded perfect. And I started pursuing that. And I was like, okay, the first thing I was like, okay, CPU, I know what it is. The basic computer we have in the lab, that's kind of an ancient computer, has a CPU. The paper said, well, it's possible to install the program in the CPU. The problem is when you are training the network with that. So my natural follow-up was to tell my advisor that we needed to buy a GPU for the lab. And it was like, well, that's not happening. We don't have funding for that. And you would have to justify why you're gonna need a GPU. And I was like, okay, so I got stuck right there. How can I solve the problem? That's when, well, Hussein, I don't think he's here now, but anyway, Hussein, he assists the psychology department at PCNEC, the Psychology Clinical and Neuroscience Center. And he told us about Garcia. And when I learned what the A meant, I was like, I don't have a background in computer science. Advanced, I don't know how to use that. That's not gonna work. Well, so he encouraged us, and one of my friends who was more sold on the idea of going to the workshop, so she convinced me to go to the Garcia workshop. So I did, and I was like ready to say, I'm gonna go, and I know it's not gonna be for me, I'm not gonna understand anything. But turns out I felt so welcome and everybody was so helpful that I just wanted to do more. I was like, okay, this is exactly what I need. So then it was the pandemic because I did the workshop in January 2020. And I didn't ask you to put your picture, but you were on the, Matthew is right there. So pandemic hit. And even though it was an awful time, it was also great because everything was shut. We couldn't go. The labs were closed. We had to basically, we had to sacrifice our animals because we didn't know how long we would be away from campus, and we didn't have a justification to just keep the animals there. And plus, nobody could come to treat the animals. So that was the perfect time to learn more about that paper, those terms, and how to use Garcia. So here in the picture this is early 2020. Matthew on a Zoom call with me, trying to have deep lab cut set up at Xena. So we got that done. Initially, I think we used Wheeler. I think it was the first one, but the problem was there's one part of the program that I need to do. The software will extract the frames from the video, and I need to annotate the frames. And Wheeler wouldn't do that. So I had to do everything in Xena, and then it worked. And so I was determined to prove that we needed new equipment for the lab. So what I did, I had remote access to my lab. You can see this is March 23, 2020. That was the day that the governor announced the lockdown. I put this sign on my computer saying that in the lab computer for nobody to touch that computer because I was remotely accessing it from home. And my goal was, I was like, now that I have everything working on Xena, I am going to run this on CPU and on Xena to prove that Garcia is better. So can anybody guess the result? It took 17 days to run in the lab computer, 27 hours on Xena.
Where you're going with that?
Yeah. So, and I did it. So it worked well. I was afraid the computer was going to crash halfway, but it didn't. It got to the end, but it was 17 days. So anyway, that's what I needed to prove that, first, I was able to overcome some barriers in like using and learning some terms that I didn't know, using the system that would help in our lab and convincing like the people in my lab that we could use those resources on campus that until then, nobody knew how to use, nobody knew that was available, and it seemed to be so distant. Now, not anymore. People know what it is, and we're, planning on using it more. So I'm going to show you this research. Like I said, we had to stop it for a while during the pandemic. And because this was not part of my dissertation for a while, I stopped doing it. So I got to the point where I was able to successfully use the Garcia system to run that software and it was so interesting because back in 2019, everything was command line. So it was a very steep learning curve for me, but, was able to do that. Nowadays, if someone is going to use that software, it's more like point and clicks, more user-friendly. But I'm glad I was able to do that because that kind of opened through the workshops and working with Matthew and with Hussein and other people from Garcia. I was able to learn a lot about, this is all Python based. I did not know until I read that paper. Python for me was the snake, that's it. So, I was able to learn some of that and got some of the results. So here, like I said, this is not finished yet. We're still collecting data, so I don't have any results that I can show right now. But what matters here is how we're able to use the software to track the frames and to eliminate the need for someone sitting in front of a computer with a stopwatch. So here is a frame of one of my rats. And let's see how I can start this. So this is what the software does. So it tracks, I can just from the frames select some parts of the animal. This is all customized for my needs. So I decided what I wanted to do. With this, once this was successful, I actually built this apparatus because the traditional way they do an overhead camera, and because I want to do more than just the traditional measure, I actually built an apparatus that has a clear bottom, and I record from the bottom, because this way I can see the paws of the animal. And in addition to just doing the discrimination index, I can do more behavior measures. So basically, like when using the Garcia Center, with that, I was able to modify the task and expand the measures that we were able to do. And yeah, instead of doing the stopwatch thing, each one of these dots are the dots of the animals, the ones that I tracked. And for example, if I want to see if the animal is looking, the direction the animal is looking, I can just isolate to the nose, for example, and I can tell where they were looking. So it's not just how many seconds they're spending with an object, but how they're interacting with the object. So it's how this system, we're able to go beyond the traditional measures. So yeah, that's basically the research I do. I mean, one of them, and this was an application of my research in like using Garcia and being able to not being afraid of computer science terms and being able to expand, what people have been doing for decades and just, like, oh, that's what you do. And being able to move on, right, to what people think it would be the traditional. So anyway, thank you, Matthew, Hussein, all the Garcia people. These are people from the psychology department. That's me and my friend Tegan, who basically forced me to go to the Garcia workshop. This is doing the Garcia tour there. And yep, that's it. Thank you all. Does anybody have questions?
You had to do labeling for it, I'm assuming, like you went through videos and you had to be like, this is the nose or the tail for frames.
Yeah, so the first step is like I have a sample of videos, it extract a subset of frames, and then I annotate each one of those frames, and then it train the network and apply it to the whole subset of videos.
How much do you have to label? How much time?
Well, it depends. So there's a likelihood test. So you start with 30 frames. and then you run on a subset, and then you check the likelihood for accuracy. And then if it's like below the threshold, you put more frames. Or if it's like within the threshold, you can just run it to the rest of the videos, you know. So this solved a lot of problems, because one of the problems that I didn't mention with the softwares that you buy, there's a problem with contrast. So for example, if I have my animal is black, if I have an object that's also black, some of those softwares, and one that we had in the lab, that would track the object. And you wouldn't know the difference between the object and the red. And with this, it recognizes what's moving, actually. So you can actually isolate the points that you don't want it to track if it's necessary. But, you know, it's pretty accurate to what's moving on the screen.
How much of your life did you save?
A lot of time. When I was thinking about that, I was like, I probably spend more time learning about all of this than tracking, but tracking is boring, you know, because, and this is like, from now, it's like, it was a little bit of time like learning, because like I said, for someone without a background, it took a while to understand some basic terms that I would know if I was like a computer scientist. But from now on, it's not, I don't worry about, we don't need to have several people scoring it. I mean, it's on the computer. You can watch the video and see if it's accurate or not. So from now on, every recording that we have the system set up and it's just running, you know, so it's saved a lot of time. And not only that, but also expanded the way we do the measurements, from like just a small subset of measurements to, I mean, the sky's the limit, basically. Oh.
How to distinguish at what stage the mouse is training, at what stage the mouse is playing with the toy? I suppose they are very similar.
So if they're playing with the toy or what? Sorry, I didn't understand the first thing.
You basically divide this into two stages, right? For the mouse. One is learning about the object. The other place is kind of like playing with the object. How do you kind of distinguish at what stage it's called learning and what stage is?
Yeah, so basically, like for the discrimination index, what's the traditional measure, we're not, that index won't tell you how the animal is interacting with the object. There's a radius around the object. If the animal enters there by accident or by playing with the object, that counts as time. So it doesn't discriminate how it is interacting with the object. And that's why, by reversing the apparatus with this new apparatus that I built in the lab and recording from below, I can actually extract more of those behaviors, just if the animal is engaging with the object or it's just like accidentally running into it, if that makes sense. You know, yeah. All right.
I want to say that, so it's difficult for high-performance computing centers to, through statistics, talk about the impact we have. I showed you some bar charts, et cetera. These stories, like exactly your journey, is exactly why Garcia exists. And the story you just told is why all of us at Garcia do our jobs, right? We love our jobs. And that was a perfect talk to illustrate why we love our jobs.
Thank you. Thank you for being so supportive. Thank you.